Jim Cornell from the Biotech, and sometimes there's an interview that we do that's just a bit long for the regular podcast. So we're going to start an occasional series of podcasts where we look a bit more in depth at a condition and what the current state of affairs is in treatment. And so in this first edition, we take a closer look at Alzheimer's disease with a conversation with Sharon L. about current drugs, clinical trials, and the pipeline. Maybe the best initial place to start would be to ask a little bit about your role in Alzheimer's and what your company does.
Yeah, I started in the Alzheimer field actually shortly after I took a position at Eisai in the US. Eisai was a startup in the US. I was the sixth person hired. So it was relatively small, the number five, six pharmaceutical company in Japan, mostly vitamins and skincare products, like a lot of Japanese companies, ultra pure vitamin E and that type of thing. But the CEO at the time, Haru Onaito, and he's still the CEO, had a vision to be bigger, and he knew he had to go to Western markets to make that happen. So he considered New Jersey to be the medicine chest of the world, since so many pharma companies were in New Jersey. And I was in New Jersey at that time then also. And he opened up their first office in New Jersey. And it was a startup with deep pockets. But as I was contemplating whether or not to move to such a small place, and at that time the quality of Japanese data was not particularly good, I thought about what's new and different, what could I learn? And I saw in their pipeline they had this drug they called E2020. And it was a cholinesterase inhibitor, slightly different from some that were already in testing. And those drugs in particular, I really believed that they should work. And so I just made the assumption. I was young and rather impetuous and fearless, and said, well, someone has to be doing something wrong because this should work. Look at the mechanism of action. This should work. So let's find out, why are these other studies failing? What's going on? How are people measuring this? I was promised if I took care of their proton pump inhibitor first and got it off clinical hold, that I would get this other project. So I got the proton pump inhibitor off clinical hold, and they gave me the project. And working through that time, you do find out a lot about Alzheimer's disease, a lot about the diagnosis, a lot about the care. But I started working in that area probably 1989, 1990. From the very beginning, as people would formulate ideas about how studies should be done, only to learn later that that's not the way the studies should be done. Selecting certain outcome measures, only to learn later why those outcome measures weren't successful. So I had the opportunity first to watch the companies that preceded Eisai as they failed miserably, and also watch the agency, the Food and Drug Administration, evolve in the way they looked at it. What's important? What do you need to see in order to approve a drug? So that was very educational. It was very interesting. Some of the early advisory committee meetings were just a circus, so it could be amusing. But that's where I started in the area and followed that through. And I ended up taking E2020 from the very first dose in humans to worldwide commercialization, something you don't generally get a chance to do in a pharma company. Usually you're in for part of it, but not all of it. But this was a great opportunity. And I've stayed with that work in some way, shape, or form since then. You learn a lot about the patients, what their needs are, what their concerns are, and how regulatory structures, by and large, they're there to approve the drug. And so they have their outcomes for approving the drug and ensuring its relative safety. But they're not necessarily there to look at all the things that are important when treating and managing Alzheimer's patients. So the cholinergic drugs, cholinesterase inhibitors, and the cholinergic hypothesis was the first one that actually showed a clinical benefit. People were already at that time jettisoning off into beta amyloid and tau and protein accumulation. And they were doing that based upon early autopsy data of Alzheimer's patients. And that's where the whole amyloid tau hypotheses were born, were based upon autopsy data from Alzheimer's patients. But just like when you go to your GP's office and you get some blood work done for glucose or cholesterol or whatever else, you know that when you're looking at your report later that there's a range of normal values. And you try to see where you fall into the range of normal. Are you up? Are you down? Are you in the middle? And everything is taken into context about not how only that analyte works, but other related analytes in your blood. And so a total picture is put together. That never happened in Alzheimer's disease, and it still hasn't happened. There's this assumption made because beta amyloid plaques and tangles were in Alzheimer patients' brain that was the cause. But the fact is, you and me, your neighbor down the street, everyone, we all collect amyloid plaques and tangles throughout our entire life. We also collect something that's called polyubiquitinated proteins, and that's a lot of syllables. But basically, when we have proteins that are old or deformed, just from use and no longer necessary for activity or incapable of providing the activity they were synthesized for, our body tags them with a molecule called ubiquitin. And that's sort of like a sign that says, okay, body, time to clear this protein. And as we get older, our ability to do that is less effective also. So we collect a lot of old and fairly gnarly looking proteins that aren't effective, that are tagged for removal, but our removal systems are just not as effective as we get older. This is, many reasons why tau and beta amyloid also tend to accumulate. We are just not so good at clearing those proteins. So the fatal, I don't want to call it the fatal flaw, but a flaw in a very complicated hypothesis, because I certainly wouldn't want to minimize the complexity of this disease. In fact, I want to highlight it, is that when you look across brains, you will see patients who die with massive plaque and tangle loads in their brain, and they are perfectly cognitively intact. There might be some hesitation here and there, but that's the same way that we hesitate when we're lifting something heavy or trying to go across, sort of skim across a very narrow curb that we want to tightrope across. We're not as good at those things as we're older as we used to be later. That doesn't mean that we're infirm or have some kind of deficiency. It just reflects the state of our bodies. So you can have all of this and be perfectly cognitively normal. And yet other patients with severe Alzheimer's disease on autopsy can have a relatively modest plaque and tangle load, but be severely demented. So there's more to it than that. There's a certain resilience factor. And we believe at our company, and I believe from all my work in this area since around 1990 forward, that neurotransmission is the bigger part of it. And so you think about ways, okay, what parts of neurotransmission and what does that mean? And those are things that are critical to understanding. And of all the mechanisms that have been looked at to actually upregulate neurotransmission and improve it, cholinergic mechanisms remain the most successful. It's just when you go through that pathway, we have cholinergic neurons all over our body. It's a master controller in our body. So if you're going to tinker with that particular system, you need to do it in a very selective way. Otherwise, you're going to get unwanted peripheral symptoms. So that can be nausea, that can be diarrhea, that can be motor dysfunction. It can be a lot of different things. However, the more selective your action is for what's going on right up here, the more likely you are to get the cognitive benefit that you want. Does that make sense?
Yes, definitely.
Okay, so when people finally have a reference range for beta amyloid and tau accumulation, then I'll be more willing to take a look at it and say, oh yeah, I understand what it is. But until that time, I'm not willing to say this is the cause of Alzheimer's disease. I'd say it's one of many causes because this is a very complicated disease. And we start accumulating these proteins, Jim, when we're probably in our 20s or 30s. So this is happening just over the normal course of aging. And everyone always talks about X is associated with Y. This is associated with this disease. This is associated with that disease. Well, quite frankly, living is associated with dying, because during the course of our lives, our bodies change. They become less effective at some things, a little more effective at other things. And we have to untangle the more complicated story to actually deal with the genesis of Alzheimer's disease and why some people succumb and some do not. So do you have any sort of questions so far about how I sort of got into this area?
No, not at all. It was interesting what you were saying. My father had Alzheimer's, and it's one of those things where I think if you get, I don't know, if you've got heart disease, it's very similar between people. Whereas it seems that with Alzheimer's, two people that have it can have wildly different symptoms and different progression. I know with my dad, it was very quick.
Yeah.
And for other people, it's not. So as you were saying, it's much more complicated than I think a lot of people think when they just hear Alzheimer's. They just think that that's what it is and that it's all the same, whereas it really isn't.
You're exactly right. And that's also why then people become disappointed when a pharmaceutical company tries a therapy and it doesn't work, or they try the same path like the monoclonal antibodies for beta amyloid. There have been failures in that field since roughly around 2006 with a company called Elan, which was a company that was founded in Ireland, actually. But Elan made an antibody to beta amyloid. So basically, it was like vaccinating oneself against beta amyloid. And they started testing it in patients with sort of a moderate dementia. And their trials failed miserably, not because they weren't getting beta amyloid removed, they were getting a lot of beta amyloid removed. The monoclonal was remarkably successful, but at the same time, there were other things that had gone on. When you remove the beta amyloid, you get sort of like a Swiss cheesy effect in the brain. You're not recreating the connections that are necessary for solid communication in the brain. So they were finding the drug did what it was supposed to do, but no one's cognition was improving. No one's function was improving. And on some follow-ups that have been 14 to 20 years later, the patients died of Alzheimer's disease at the same rate that everyone who had never been exposed to the drug were dying. So this is why companies in the later years, so the Elan stuff started coming out around 2006, 2007. And so the initial gold rush was, okay, none of these drugs work. It's a waste of money to develop them. Throw out the baby with the bathwater, that whole kind of thing. And it's never that simple in pharma. And something that's complex, it's never that simple. But what happened in Alzheimer's disease too was that because this was where the focus was, where everyone wanted a cure and no one wanted a treatment that wasn't a cure, the whole field of Alzheimer's got a reputation. Everything was in one bucket, that this is a black hole for money. Don't invest in Alzheimer's disease research because the drugs don't work and it's just a total waste of money. So if you were working on another pathway, like I tend to be in symptom management, not cures, but symptom management through the lifetime of the disease, it was quite difficult to get funding, and because investors would be gun shy and say, no, it's a waste of money. And certainly the billions of dollars it takes for a monoclonal antibody project, that's quite daunting. Working with small molecules and symptom management, that's much easier path to follow, much cheaper, and you can get an idea if your drug is working instead of in 18 months, in around 12 weeks if you've got something that's going to be helping those patients. Does it change the course of the disease? It depends on how you define that. Because what we do know and why we try to keep patients active is that the more your neurons can fire, the slower the disease progresses. It's just like when you use your muscles to exercise, your muscles stay toned and more capable for you. It's the same thing with your brain. So yes, Alzheimer's patients are encouraged to socialize, encouraged to exercise, encouraged to do crossword puzzles, a lot of things. Anything that makes your brain's neurons fire is good. And the more you fire, the more you can sort of ward off having the symptoms of Alzheimer's disease take a really good hold on you. Can you do it all the time? No, because as you learned, it can be very, very different across patients. And that's because there are other confounding variables, and we don't know what all of those are yet. It's so tragic people hope for simple answers, and it's just not that simple.
Sure. Do you think that one of the reasons why investment hasn't been as good as it can be is that there's a little bit of a perception of, well, everybody gets old, everybody has issues as they get older, you forget stuff and generally you decline and your brain declines too. So do you think that maybe there's a bit of a kind of a thought that, well, we get old anyway, So we can't cure aging, so why worry about it?
Well, you sort of tapped into something that is on my mind a lot. And I'm going to regress back a little bit and think about how much the monoclonal antibodies cost to give. So with Aduhelm, it was $56,000 a year, which was just crazy aggressive. With lecanemab, it looks like Eisai is going to go for around 23,500 per year. But then there's all the things that need to go into giving infusions and monitoring for ARIA and all these other indirect costs. So that's highly expensive. And so with an old person, sometimes people are not willing to look at what turning back the clock 6 months, what the benefit is. In oncology, even when patients are a bit older, the majority of the patients are more middle-aged and more productive. And so when you can increase survival, not functionality, but survival for 1 month. In oncology, that's really celebrated. That's, wow, this is great. And we improve survival by 1 month. And yet, when you have an old Alzheimer patient and you've turned back the clock, say 6 months, and their ability to function, maybe kept them out of a nursing home for 6 months, maybe kept skilled nursing care out of the home and eased the burden on the caregiver for 6 months, that's not celebrated. So, I would like for someone to explain to me why that difference. And is it because old people tend to be, at least in Western societies, not in Eastern societies, but in Western societies, older people tend to be dismissed more quickly? And yet you look at someone like, if you know who Anthony Fauci is, much maligned as he has been by certain people on the right. This is a man who is well into his 80s. He's almost 90. And I would put him up against anyone as far as he is with it. He is sharp. He mentally can dissect things just as well as he could when he was younger. And he's a terribly effective communicator and a valued resource in our lives. Not everyone's an Anthony Fauci, but at the same time, there are people who can live good and productive lives more independently, enjoy their families. Is there no value to that? This is the question I ask, because I don't want to get to that point and have people willing to just shove me in the chair in the corner, sedate me so that I'm not agitated and delusional. And that can be just a matter of being demanding or asking questions or saying, I don't want to do that right now, and taking away someone's agency. Because when we are ill, this is what goes first, is our agency as people, as human beings. That gets taken away to some extent by the healthcare system. And so when you're old and have some mental faculty issues too, agency is taken away much more rapidly. And this is why you reach this point where all of a sudden the Alzheimer's patients sort of fall off a cliff and die. It's because they can't fight for their own agency. There's no one there to fight for it for them. There was a great study done in Scotland and England. This is called the Domino study. And in the Domino study, they had community dwelling individuals with Alzheimer's disease. And it was severe Alzheimer's disease, but they could still be in their homes. And so they started this study to see, okay, all these people right now, they're on Aricept and sometimes some other drugs too, but at least they're all on Aricept. What happens if we decide, because they're severely demented, that the Aricept doesn't matter anymore? We'll take that away and then see how quickly these people end up in nursing homes where people die very rapidly thereafter. And in this study, they did what's called a Kaplan-Meier survival curve. I don't know if you're familiar with it, but there will usually be a curve, and this will show the usual rate of progression, where the outcome is, in this case, nursing home placement, or sometimes the progression, since it's survival, can be death. And so they had two survival curves. They had the patients where they took away their Aricept to see how long they could survive outside a nursing home. And then they had the people who were kept on Aricept to see how long they could survive outside of a nursing home. And patients in the non-Aricept group started their attrition process quite a bit earlier. And those curves of survival did not cross paths where they were the same until 42 months into the study. And the average amount of time that they were able to stay home with their families and be manageable was around 8 to 10 months on average. Some a lot more since the study went out and it didn't cross over till 42 months. Some obviously probably went to the nursing home earlier. But I look at that and I say that means something. It means something to the relatives, the family. It means something to the patient. It means something to healthcare costs, because in-home nursing care and nursing home costs are the major drivers of costs in the healthcare system. So everybody wins by taking a drug that in the EU you can get for less than $1,200 a year generically. So why would people not do that? Why would people be so willing to just sort of ship them off and turn out the lights and not pay attention to them anymore? That says as much about us as it does about our healthcare system. And so you asked a very sensitive question with that, but I think it's a question that we can't ignore. Ageism exists. I'm getting older all the time. Things aren't wanting to work the way that they used to. I find it frustrating, totally frustrating, where you wake up and something isn't quite right and think, really? Seriously, this is going to happen now, or I'm getting a slight bit of essential tremor as I get older, and so it makes putting on eyeliner really a challenge. It's like, stop, stop this already. And yet, essential tremor is a part of aging. There's nothing wrong with it. I can't really treat it. It's just something that I'm going to deal with. But does that make me a frail person or a lesser person? No, because I can still go out and run a marathon if I'm trained for it. So I don't want to hear about that. But the view of younger people looking at the elderly with some evidence of being elderly is not quite so charitable. And I think we have to recognize that. And it makes me sad.
It's not related to Alzheimer's, but the BBC had an interview with somebody that was saying that older people find it much more difficult to get jobs. And in spite of masses of experience, as you said, I think ageism is often subtle and often not even recognized.
Yeah, because the people who have it are younger and they're not seeing ahead. This month in the US, more than 500,000 new jobs were created over the last quarter, and that's astounding. And a lot of these jobs are going unfilled. We've had the lowest unemployment rate now since 1969. You cannot tell me that a lot of these jobs could not be filled by older people. And yet there are so many hurdles to doing that. Even just the attitude of someone who goes into a sandwich shop, to order a sandwich for takeaway, they're going to be looking at the person on the other side of the counter and make decisions about how well they think that sandwich will be made. So now if we go to my company in particular, like I said, I like cholinergic processes. Where they're limited, again, was that selectivity for brain activity. So my company it works in conjunction with a drug like Aricept. So what our drug does is it stimulates what's called a T-type calcium channel. Calcium is probably one of the most important molecules in our body. It's an ion that turns on and off so many different physiological activities. But this type of channel is very interesting because most of the channels are sort of like on and off switches. You open the gate, you close the gate. This is a very subtle, low voltage gate. So the effects that it produces are subtle, and they're more into biochemical messaging within the cell. And our drug modulates this channel. And what it does is it allows a little more calcium to flow into what are called presynaptic neurons. When that happens, neurotransmitter that's floating in vesicles in that area. Should I put up an illustration so that you can see what I want? I can do that. So we modulate this T-type calcium channels. Now those calcium channels are up in this region. This is a presynaptic neuron, and this is a postsynaptic neuron. And in between, there's a cleft. And so what happens is that the transmission, the discussion between two neurons, what's going on in this neuron has to jump across this cleft to the other side to keep the conversation going between the neurons. So we act at this particular step on this channel, and by modulating it, we can allow more calcium to come in. These little blobs you see here, those are called vesicles, and inside those vesicles are neurotransmitter. And in this case, the neurotransmitter is called acetylcholine, or ACH. So these vesicles have to migrate down to this membrane, fuse with it, and when they fuse with it, they release their contents into this cleft I was talking about. And from this cleft, these neurotransmitters need to sort of jump across to the other side, bind to a channel on the other side that's called a ligand gate, open it up, and sodium, extracellular sodium, floats in, and that keeps the nerve impulse going. So Aricept actually works inside this cleft. We have naturally occurring enzymes that float around in our body. They keep our neurotransmission very exact so that we don't overstimulate ourselves. So have you ever used a insecticide spray on a bug and watched it die? Really quite shocking, and people never think about how it happens, but usually right before death, the animal is twitching, or the insect is twitching and running around and acting very crazy. And that's because the insecticide contains a cholinesterase inhibitor. And it's basically just overstimulating the insect to death, which is quite shocking when you think about it. It's a horrible thing to do. So we have naturally occurring enzymes that keep us from overstimulating ourselves. Donepezil, or Aricept, works in this cleft, and it inhibits that enzyme. And by inhibiting that enzyme, it allows more of the neurotransmitter to cross the cleft and maintain the nerve impulse. We work up here, and we cause more of the neurotransmitter to be released. We're increasing the release of these. And so we're putting more of these out here in the cleft, plus in the presence of donepezil, what we put out there is preserved. And so we have increasing the release of the neurotransmitter, preserving the neurotransmitter, and getting additive neurotransmission, additive improvement. Okay, and here's why it makes sense in an Alzheimer brain, because this is nice and clean and very pristine, but that's not what brains look like, especially when we have plaques and tangles and polyubiquitinated proteins. What instead it looks like is this. So here's that presynaptic neuron, there's that cleft, and here's the postsynaptic neuron. And all these aqua colored things, this is protein that's just stuck all over the place in our brain. And so that neurotransmitter is trying to jump this cleft, but it's trying to do so with all types of interference. There's also interference on the pores that cause natural release that are on both sides of the synapse. So this is what we're trying to overcome. And this is why Aricept was effective initially, is because it preserved the neurotransmitter, gave it more time to get through all of this garbage to the other side. And what we're doing is increasing the release on this side, plus taking advantage of Aricept's natural action to allow more to get to the other side and improve neurotransmission. What that does is it improves both cognition. And when you improve cognition in a patient, you also improve their global function, their day-to-day functional capabilities. And so in our phase 2 studies, what all the regulators want to see is they want to see two things in addition to safety. The first one is that any treatment for Alzheimer's disease has to improve cognition, because that's considered the core symptom of the disease. But they want to know that if you're improving cognition, that it leads to a meaningful improvement in day-to-day life. And so that's where you get improvement in function. And you have to meet both of those criteria in order to get approval for efficacy. And then, of course, you have to have safety. So that means a sufficient number of exposures to elderly patients for a sufficient period of time so that you can understand the risk benefit. So in our phase 2 study, this is precisely what we did. We thought, could this drug work without Aricept? We thought, yeah, maybe it would, but what Aricept does is pretty important. So we're interested in the combination. And combination therapies are used in diabetes, they're used in heart disease, they're used in Parkinson's disease, but people are using combination therapies in Alzheimer's disease. They just don't seem to think of it. But I like cholinergic mechanisms because they are proven to be effective. And so we did phase 2 study, randomized, double-blind, placebo-controlled, which is the only way I like to do these studies. That's the only way you get honest assessment of your drug's properties. And in phase 2, we found out, yes, we improved cognition on the Alzheimer's disease assessment scale, cognitive subscale, which is the gold standard. And on the global, we use the Alzheimer's disease clinician's global impression of change, which was created by the Alzheimer's Disease Cooperative Study Unit. It too is a gold standard, and it's one of the hardest hurdles to breach because the person who does that assessment is siloed from all other aspects of the study. They're not allowed to know about cognitive test scores or about adverse events or about abnormal labs or anything. All they know is they evaluate the patient in an intake interview, get an idea of where their deficits are and their function and their ability to interact with their family at home. And then they reassess that at scheduled intervals during the study without getting any additional information. So we also doubled the proportion of patients considered to be improved in only 12 weeks. So our drug, we know, is going to do what we want it to do. The thing now that we have to do, our next step, is to get it into phase 3 trials so that we can look across a broader range of patients and collect all the safety data that we need to go forward. And because of the subtlety of T-type calcium channel action, the safety profile is really very good because this is selective for the brain. So all of these peripheral effects that some of the other cholinergic agents cause, we don't cause. Everything is very selective. I remember in phase 1 where we're pushing the dose to see, okay, let's see the adverse events. I got very nervous because we weren't seeing any adverse events. And so I had the analytical laboratory do a blinded analysis of the plasma samples so I could be sure that drug was getting in and being absorbed and that the amounts that were getting in were proportional for each increasing dose I gave. And both of those things held true. It's just the selectivity of this action and the subtlety of it is enough to improve cognition and global function, but not enough to cause events that would preclude additional use of the drug. So that's where my company is. I like anything to do with calcium. So in our pipeline, we have some earlier agents that work through a number of enzymes related to calcium. Calcium is also very critical to what's called autophagy. And that's our innate ability to get rid of those polyubiquitinated proteins. That's auto-eating is basically what it's related to. And it turns out that there are T-type calcium channel mechanisms involved in that as well. So we have this direct effect on neurotransmission, and there may be some indirect effects that we can investigate later on these protein processing issues. But the main idea is to take the benefits of it, improve cognition, improve global function, and get that out to patients. So they, like in the domino study where, the paths crossed at 42 months, well, wouldn't it be great if we could increase that time to when the paths cross to 52 months or to 60 months, where you're keeping people out of a nursing home?
How does the level to which somebody has Alzheimer's make a difference? I mean, clearly, when my dad was declining very quickly over a 6-month period, I don't think anything would have helped. Is it the case that the earlier that you're able to intervene, the better the results are going to be?
That part remains to be seen, and I don't know all the components of your dad's Alzheimer's, if he had any mini strokes or any other protein deposits or frontotemporal lobe involvement. Those can all impact the course as well. Or he could have just been resilient as anything for as long as he possibly could before he finally just couldn't take what was happening in his head anymore. So without knowing the specifics of the patient, these drugs are meant to be used over the lifetime of the patient. So even Aricept, mild, moderate, severe, you really want to be going across the full breadth, because there is evidence with Aricept even, and I know because I published this work, that, again, I said using your neurons is helpful. Aricept in and of itself will preserve the volume of the hippocampus over time. And the hippocampus is the seat of learning and memory for us. So again, anything that keeps your neurons firing has this ability to provide benefit for longer. But when you listen to what's in the popular press, You don't hear this. You'll hear people say, oh, yeah, Aricept, it works for about 6 months or so, and then the effects go away. And that's simply not true. It doesn't stop the progression of the disease. The disease is relentless, it's going to keep going. And so people will get worse. But is the benefit of the drug gone? And the answer is no. And so that's one thing that the domino study showed. Yes, these people were continuing to progress, but they were able to stay in the home and be functional longer and put a value on that. And it's usually someone very young who says it
Mm.
works for about 6 months and then the effect goes away. And I've never said that. I mean, I got so passionate about this area when I was working in it that I've done a lot of startups and a lot of other pharma work and a lot of the business part of pharma. But this is like a magnet for me because I feel like we have a lot of unfinished business there. And I also truly do believe, as you have an inkling, that if this were happening to a young person, that people wouldn't be so sanguine about letting them just go off and die.
How important is early diagnosis? Is that a key to being able to treat it more efficiently?
I think so. And for all the monoclonal antibodies, those are prodromal and pre-AD. So early diagnosis, I think, is important. It's very helpful because people can know what's happening, can try to work with agents earlier and prolong function as long as possible. But quite frankly, most patients don't get diagnosed until they're in the early stage of moderate. And so this is just sort of our healthcare system in general. People will report to a family physician because they're having real issues. And so they're going to be at the more early moderate stage there. But that doesn't mean that you don't start treatment and keep treatment going, because whatever you do, the intervention is going to make it better than it was before, unless the dementia is caused by stroke or B12 deficiency or thyroid disease or some other cause.
There was a lot of mainstream press about 6 months ago or 5 months ago, or whenever lecanemab was kind of being touted as the great new drug for Alzheimer's. What do you think the future holds, I guess?
That's a good question. And I will let you know that I used to work at Eisai. And so I know these people. In fact, when I did my phase 2 study with our drug, they gave me all the Aricept I needed for background therapy with my patients. So my relationship with them is good. And I was very happy for them with that study because if you actually look at the data, it was superbly conducted. There's no equivocation. The p values are fantastic. But what the drug does needs to be put into perspective. And in this case, when you look at the long-term data around 18 months, you'll see the same kind of thing. The slope of disease progression is slightly different. You don't know what's going to happen after 18 months. It's slightly different than on placebo. And so in this case, it's a turning back of the clock of about 6 months. But because the fact that you see a slope tells you, yes, the disease is progressing. It's just progressing a little bit slower. So they know if they remove so many centiloids of protein that they're producing a benefit that's turning back the clock about 6 months. And so then healthcare agencies have to ask themselves, okay, if they know this many centiloids of protein, people are likely to not have any symptoms for a while. Do you stop treatment then? Do you continue treatment then? It's $23,500 a year. How does that work into healthcare costs over a lifetime? All these questions get asked, and they're always asked with a slightly more pessimistic viewpoint in an old person. It would go back to that theme again because it does run across all of Alzheimer's disease. But this is a good drug. The risk of ARIA is significant. And remember that these patients in Eisai's studies were all with specialists. They were all getting scanned regularly. Their safety was being followed in a way that is not possible in general practice. It's just not going to happen. And so there's going to be ARIA that are undetected, where you can't withdraw therapy because people don't even know it's happening. The other issue, too, in an aging population with a drug like that is that when there looks to be a stroke, and patients go to the hospital because it appears as though they have had a stroke. Most strokes are what are called dry strokes. They're ischemic strokes. Very few are hemorrhagic strokes. But if the patient that you're treating perhaps as having a hemorrhagic stroke due to ARIA, or if they have brain swelling and microbleeds due to ARIA, that may not be known because the treatment for ischemic stroke, and you want to treat as soon as possible to preserve oxygen flow to the brain, is an anticoagulant, is TPA or streptokinase. And so by definition, if there's some bleeding or vessel weakness in the brain that's related to ARIA, you're going to end up with a situation on your hands. That's just there. And so people need to decide risk and benefit, risk and benefit. But when you delay treatment in the case of patients who have a stroke who are taking lecanemab, I almost feel like they should have some type of a bracelet or something of that nature on, so that when they are taken to the ER, that instead of just treating first, as what is the usual course of action when the symptoms are consistent and you can rule out a major hemorrhage, and that's usually through symptoms also, that maybe instead they go for CT straight away, or they get an MRI if possible straight away, and they go through the history to see, are they taking lecanemab? They ask into the history to see, has there been any evidence of brain swelling or microhemorrhage, that they can capture that data on patient presentation. Is it a manageable side effect? Sure, it is, but people have to be aware. And I think only by having some type of identifier on is the average physician in the average emergency room going to be able to address that effectively. I don't know if Eisai would agree with me on that, but Haruo Naito has a lot of integrity. Eisai as a company has a lot of integrity. And I think they want the best thing for their patients. So that to me would be a small ask. Will regulators actually mandate it? You know, the FDA approved Aduhelm, so I don't even know what to say about that, because that to me was not a just approval. That was the worst of pharma. There were almost 13,000 patients screened for those trials, those 12,800 and some patients. And then they would come into the study. Some would drop out because of ARIA, some would drop out because of noncompliance, but still you're starting with roughly 12,800 patients. And then you go through the study. There were failed studies. There were no signal studies. There were all kinds of things that happened. When they got down to the very end on what drove the approval of this drug, it was a small cohort of 289 people. So 12,800 to 289 people that maybe sort of got a benefit, $56,000 a year for treatment. No. That's unconscionable. Lecanemab is a different animal. The data are clear. The inclusion-exclusion criteria were clear. They're absolutely positive to me that they're going to be patients who get a benefit. But, and here's the but, all of these patients were either prodromal or very early Alzheimer's disease. Very early. So that means you have to catch this early. There is zero evidence to show that giving this drug to a patient with moderate disease is going to be beneficial. And in fact, if you go back in time and look at the Elan data and earlier studies with monoclonals, that data actually informs you that in the moderate stages of the disease, there's no help. And that's because you're removing the amyloid plaque and tangle, but there's still no rearborization. There's still no restoration of brain tissue. So I'm equally interested in one drug we have in our pipeline that is a nerve growth factor enhancer, because over the long haul, if you can sort of reestablish communication, fill in the Swiss cheese bit, there's the opportunity to be able to provide benefit even in later stages of the disease once you call out the plaques and tangles. That's the great unknown. And that's what, when I'm trying to relax on a weekend, thinking about the science behind that and reading the latest papers on different biochemical mechanisms, I find that quite relaxing. And that's my fun, because I don't have to think about the business parts. I can just think about the magnificence of our bodies and our brains. And if you want to know what brings people into this area, I can't speak for all people, but for me, it's being totally blown away by how amazing our bodies are and how amazing all these interlinking mechanisms are that let us function every day. There are people who have been at this as long as I have, but usually their take on it is maybe the same, maybe different. So there are probably a half a dozen of us who have been at this with patients for a very long time. And I think our viewpoint is a little bit different than the viewpoint of both investors who fund companies or regulators or the general public, because they can only, general public especially, they can only be informed by what they read. And sometimes what they read just gives a piece of the story and not all the story. Or maybe that's and through your work you can help them appreciate small advances and small victories. An example I use a lot is Parkinson's disease. 40 years ago, my old department chairman was one of the first people to use levodopa in Parkinson's disease, which made a huge difference, just replacing the dopamine. But still, if you look at Parkinson's 40 years ago, it really was a death sentence. People did not last long. By the time they were diagnosed, it was usually too late. And now we have started layering on therapies and layering on additional therapies and finding ways to manipulate things that are going on in the brain that we knew nothing about 40 years ago. And so you end up with someone who, granted, he has a lot of resources, financial resources available to him, but someone like Michael J. Fox, who clearly has Parkinson's disease. You can tell when you look at him, but you cannot argue that he doesn't have a productive, fulfilling life. He's watched his children grow up. He's been able to enjoy his family. He's been able to continue his career. And this is what happens when people take the longer viewpoint on the fact that you make incremental steps, and those incremental steps continue to add up. So I'm hoping that by making incremental steps in improving cognition and global function, that we allow people to have their life. We allow their families to enjoy them. Caregiver burden is tremendous, and we're having far fewer caregivers because birth rates are down. So all of these things all go together.